Our next speaker is Janusz Pętkowski, who's from the University of Wrocław in Poland, and he will tell us about the possibility of biochemistry in concentrated sulfuric acid. Thank you.
Thank you very much for the invitation and for the kind introduction. And what am I going to talk about today might sound like a complete science fiction, yes, because we are going to talk about the possibility of biochemistry in concentrated sulfuric acid. But I hope that, towards the end of this talk, you are going to look at this unique and aggressive solvent through much more favorable lens. So, of course, we are interested in concentrated sulfuric acid because of our neighboring planet Venus. It has these mysterious clouds that cover the entire planet, and as far as we know, the main constituent of these clouds is concentrated sulfuric acid. And of course, the surface of Venus is actually completely uninhabitable. It is 465 Celsius degrees at the surface, but approximately 50 kilometers above, you have this so-called temperate zone or temperate cloud decks. And why it is called temperate is because at the bottom of the clouds you might have maybe 100 degrees Celsius, and at the top of the clouds maybe, let's say, around 0 Celsius degrees. So this is sort of a range of temperatures in which you actually could expect some complex organic chemistry, not to mention very speculatively, hypothetically, lights. But of course, the devil is always in the details, because these clouds are, of course, composed not of little droplets of water, as we have here on Earth, but of concentrated sulfuric acid. So the dominant liquid on Venus, on this planet, is actually very, very different. And this actually brings us to the following point, that the environmental challenges for life in the Venusian clouds, if we actually speculate about that, are absolutely unique and exceptional, because this particular concentrated sulfuric acid environment actually makes our own life here on Earth impossible to exist in this cloud environment. And this is because this concentrated sulfuric acid is actually many, many times more acidic than even the most acidic places that we have here on Earth. So even these very alien-looking Dalol pools in northern Rafah in Ethiopia are still, they might look like they are from different planets, but it is still our own Earth, and it is still a solution of low concentration of acid dissolved in water, and not the other way around. And of course, there is also a related problem that Venusian clouds are actually extremely dry. They are maybe 50 to 100 times drier than even the Atacama Desert, which is one of the driest places here on Earth. So this brings us to the following conclusion. And again, I chose the red font for whatever reason, and that might be not the best. But what is actually quite important is that if we actually consider Venusian life, then this has to be life as we don't know it. The only question is to what degree, because those two challenges actually make our own life, Earth-like life, absolutely impossible in the cloud environment made of concentrated sulfuric acid. But what is actually also quite important, this is something that we now study quite intensively, is that we do not want to see these two challenges as something that prevents all life from existing. We actually want to embrace this unique environment and ask a question: can we actually have some alternative biochemistry where concentrated sulfuric acid is actually the solvent instead of water? And at the first glance, you might think that this is complete nonsense and a complete science fiction, because if you actually Google what happens to, for example, sugar upon addition of concentrated sulfuric acid, and we will quickly stumble upon a very spectacularly looking YouTube videos with many likes and retweets and God knows what, that actually show you a wonderful reaction, vigorous, sort of reaction that under 1 minute produces this sort of carbonaceous blob that has nothing to do with complex organic chemistry, not to mention life. But of course, what we have to realize is that this is actually a very misleading experiment. It only shows that sugars are actually incompatible with concentrated sulfuric acid and not organic chemistry as a whole. Yes, because thanks to our work and of course our collaborators, like for example the work of Dr. Janusz and Professor Stephen Werner, we know that we can create very complex organic polymers, for example, in concentrated sulfuric acid from very simple precursors, like, for example, from formaldehyde or carbon monoxide. So even the simple atmospheric gases like carbon monoxide, which is present also in the clouds of Venus. So the existence of complex organic chemistry in concentrated sulfuric acid is essentially a fact. It's just a question how different this organic chemistry is from our own water-based Earth-like biochemistry. And we asked even more simple question that apparently nobody before really asked and thoroughly addressed. Which of the building blocks of our own biochemistry are really unstable in this aggressive solvent? And this is a beautiful work by Max Ziger when we actually asked which of the amino acids in our own proteins are actually stable in this solvent. And miraculously, 19 out of 20 of our amino acids that build our proteins are actually perfectly stable in this solvent, with the exception maybe of tryptophan. They sometimes undergo some chemical modifications, like, for example, sulfations or sulfonations, but this doesn't mean that they are unstable. This means that this solvent actually acts on them, modifies them, but not necessarily destroys them at all. So this was actually such an unexpected result that we even landed a cover in astrobiology, because it's a very, very simple experiment, but at the same time, the implications of it are actually quite profound. So we were emboldened by these results, and we asked even more outrageous question. What will happen to the peptide bond in concentrated sulfuric acid? And the peptide bond is probably one of the most important chemical bonds in our own biochemistry. It brings together two amino acids, so we can actually form proteins, peptides, polypeptides, so the executive branch of our own biochemistry. And at the first glance, you might think that this is another completely ridiculous question, because this red bond here, the peptide bond, probably will completely fall apart in concentrated sulfuric acid. So we tested this. And actually, we were absolutely surprised, because there are some, and this actually some just for example of four dipeptides, so probably the simplest peptides that can exist, built just from two amino acids. And as you can see, some of them are completely stable in this 98% concentrated sulfuric acid for periods of many, many months. And this is, for example, glycine, and adenine glycine dipeptides, very simple ones, but nevertheless, we would expect them to break. But they do not. Some of them, they do indeed react in this solvent, like, for example, alanine. So we embarked on a quest to actually understand this solvent better. And we asked ourselves the question, why some of those peptides are actually stable and others are actually reactive? And this is a good moment to actually remind ourselves that diluted acid is actually a completely different substance from a concentrated sulfuric acid. Because in diluted acid, of course, all of those dipeptides undergo this canonical textbook biochemistry reaction that is this acid-catalyzed hydrolysis. So acid catalyzes the reaction that allows water to break up the dipeptide and release the individual amino acids. In 98% concentrated sulfuric acid, something else happens. Some dipeptides are completely stable, and the other ones, when they do react, actually release different products. Like, for example, this alanine amide here, so amide modified alanine or acrylic acid. And a different bond breaks, this bond between alpha carbon and the nitrogen, so not necessarily the peptide bond itself. So we wanted to understand what is actually going on, why this alanine dipeptide actually breaks down and forms this rather amber-looking solution. And we hypothesized that what is actually important for this reactivity is this carbon-hydrogen bond here on the side chain of the alanine. So what will happen if we actually switch this carbon-hydrogen bond to something that should not react in concentrated sulfuric acid? So we did this after many, many months on trial and error, and a lot of thousands of dollars spent, because this was actually quite difficult to synthesize and many companies actually failed. We created this fluorine variant of alanine dipeptide. And what happens is that this indeed is a completely stable dipeptide in concentrated sulfuric acid. So we were able to stabilize our peptides essentially. We have a path towards stabilization of peptides in concentrated sulfuric acid. And this is actually completely unexpected. You would not predict that. But yet this happens. And in parallel, of course, if we go through our own biochemistry, we asked ourselves a question: what about our genetic component of biochemistry? What about our famous letters of our genetic code, that G, C, A, and T? And they are also very, very stable. They are stable for more than 1 year. We stopped measuring at some point because it was just boring for us to measure it all the time. But, they can undergo protonation on amines and carbonyls and so on. But this doesn't necessarily mean that they are unstable. So we asked another outrageous question, and possibly also a rhetorical one. Can we actually build a genetic-like polymer that would be stable in concentrated sulfuric acid as an example that, this solvent can support such complex organic chemistry? And yes, we can. But this is, of course, a good moment to remind ourselves that all those spectacular YouTube videos are actually correct. We cannot have DNA in concentrated sulfuric acid, and the reason why is because we have deoxyribose and also other groups that are very, very reactive in concentrated sulfuric acid, so DNA will fall apart immediately. But there is another polymer that we tested, and it's called peptide nucleic acid, PNA. And again, we were absolutely surprised to realize that it's actually stable at room temperature in 98% concentrated sulfuric acid. If you actually, and this is a little bit of a tangent here, but if you subscribe to the so-called polyelectrolyte theory of the gene, so the idea that any genetic polymer has to have a repeating charge on the backbone for the evolution to actually happen, then in concentrated sulfuric acid, you also have that. You just have a reverse charge, not a negative one like on DNA, but a positive one, like because the carbonyl groups of the backbone of the PNA are going to be stably protonated in this solvent. So we were very, very excited. We were very, very happy about this result. But unfortunately, we are not there yet exactly because this PNA falls apart in higher temperatures. In around above 50 degrees Celsius, it actually breaks down. So we are now working very, very hard to substitute this amide bond here that connects the linker and the base to the backbone with something else, and to have an example, the genetic-like polymer that will withstand the entire range of temperatures that we could actually have in the clouds of Venus. So finally, there is also another element of biochemistry that is also crucial, and it's the barriers, the membranes, the cell walls, something that will denote the, inside from the outside of the cell. So we actually forged a very fruitful collaboration with Professor Jack Szostak and his group, and Dr. Daniel Dusdevich and Colin Nisler. And we asked, can we have vesicle formation? Can we have membrane formation in this aggressive solvent? And of course, this is another rhetorical question, because yes, we do. And we actually can see both the micelle formation and vesicle formation in this aggressive solvent. And I could go through all the biophysical graphs with you, but this is, of course, very, very boring. So people say seeing is believing, but in this case, probably you cannot see anything because, unfortunately, the red dye is stable in concentrated sulfuric acid, and red dye is not compatible with PowerPoint. So we are working on that. We will try another dyes next time, yes? But what you could probably see here, is that those are beautiful vesicles here, membranous structures that form in concentrated sulfuric acid, and they persist there, they are stable, and nothing happens to them. And the red dye that allows us to visualizing this is also stable. This is also quite remarkable if you think about this. But what is even more remarkable is the slide on the right, in something that you should see, but you probably cannot, is this sort of, sometimes we see the formation of tubes, of little tubes in this concentrated sulfuric acid, and they have this purling behavior, the budding. They bud out little small vesicles. So we have an example of sort of a prebiotic vesicle-like cell division, I would say, or vesicle division, something that happens also in water, but in water it requires special conditions, special lipid composition or turbulent water flow to actually make this phenomenon happen. In concentrated sulfuric acid, this happens immediately. Either we were very lucky and we just grabbed random lipids and random conditions, and this happened, or maybe there is something about concentrated sulfuric acid that actually makes this behavior more likely. So we are actually, and this research is maybe 3 years old or so, we are now on the quest of actually formulating a new paradigm. We are not afraid of controversial topics and controversial ideas. We actually think that life does not necessarily need water, that the fact that we are water-based life is a consequence of our environment that we have here on Earth. But it doesn't necessarily mean that life elsewhere has to be based on water. Because we now, thanks to our collaborators, also know that this complex organic chemistry is compatible with concentrated sulfuric acid. We are now building an example genetic polymers. We can even stabilize peptides if we wanted to. And of course, we can also form various complicated membrane-like structures. And why do we do this? There is an overarching goal here, because we would like to go to Venus and actually study the mysterious clouds see what chemistry we can actually have there. And I have a privilege of being a deputy PI of an initiative that is called Morningstar Missions to Venus. And we have a series of missions plans and under development. And of course, this has astrobiology at its core. So we are not afraid of that. We are actually making astrobiology a focus of this endeavor. And our goal is to search for increasingly complex molecules, organic chemistry in the clouds of Venus, because we actually think that organic chemistry is going to be there. And we have a couple of missions planned. The first one is we are developing with our partners at the Rocket Lab, and this is the so-called Rocket Lab mission to Venus, which is hopefully going to launch soon in 2026. But we also have other concepts that we are developing in parallel, and it's all because we want to find potentially even signs of life in the clouds of Venus. And I will end here. Thank you very much. And of course, I will end with thanks. And there are many, many people who are involved in this initiative, of course, but the organic chemistry here is the team the research in organic chemistry is the team that I showed here. And special thanks go to Professor Sarah Ziegler, who is the leader of the entire initiative. And nothing that I showed you here today would be possible without her. So special thanks to her, but of course also to Max Ziegler, William Baines, John Grimes, our NMR expert, and also the group of Professor Jack Szostak, Daniel Tusewicz, and Corinne Nisler, and also Dr. Jan Szpaczek and Professor Stephen Penner. Thank you very much, and I look forward to interesting discussions.